Good afternoon, everyone, and thank you for giving me this opportunity. So the abstract that is allotted to me is the early results from a phase 1 multicenter trial, which looked at PSCA-specific CAR T cells and metastatic castration-resistant prostate cancer. This was a multi-center trial from various centers in the US and was presented as a poster in this year's ASCO GU conference. So just a little background, basically CAR T cells, like we all know, are T cells from a patient which are isolated and genetically engineered to express a protein that targets antigen on the tumor cell. And then these T cells further have properties of expanding within the body and then circulate and attack all the tumor cells and lead to an immune response and lead to a control in the disease. So various antigens have been looked at for as a target in prostate cancer. They looked at PSCA, which is a prostate stem cell antigen, EpCAM, and PSMA. And these three had a lot of preclinical and early phase, predominantly phase 1 trials. And these are three of these studies that are looking at PSCA CAR T cells in prostate cancer, 2 of them in the US and 1 in China. The full results, or basically the trial is still ongoing. They have not completed yet. So coming to our study that we are going to discuss today, this was BPX601, that is the CAR T cell product that is expresses the CAR to the PSCA antigen. So in addition to this antigen, the PSCA antigen, this CAR T cell had a property that it would be stimulated by Remidusib. So Remidusib is a drug. Basically, it's a tacrolimus analog, which is lipid soluble. It has multiple actions within the cell by targeting the caspase pathway and inducing cell apoptosis in normal cells. But its function in this CAR T cell is basically it targets the mid-88 CD40 co-stimulation molecules. So if you know that CAR T protein has 3 parts, the extracellular, the trans membrane, and the intracellular domain. And to improve the function of these CAR T cells, they are engineered to have certain co-stimulatory molecules which enhance the T cell proliferation and allow the CAR T cells to expand within the patient's body. So this study included the administration of this drug called Remidusib, which was supposed to trigger, it was a switch to these T cells to enhance their potency and keep them persistent in the circulation. And this was a phase 1, phase 2 study which was looking at this particular CAR T cell in prostate and pancreatic cancers. So we focus on the phase 1 dose, and this was a cell dose escalation study to look at the maximum tolerated dose. The phase 2 is not yet started. So they included metastatic CRPC patients who had failed 2 prior lines of therapy, including an androgen receptor antagonist and a taxane. And they excluded, of course, patients with unstable bone lesions, coagulopathies, cardiac dysfunction, or active or symptomatic CNS mets. The primary objective of this phase 1 study was, of course, safety and tolerability, that is to identify the MTD and dose-limiting toxicities, and to get a dose for the phase 2 part of the study, that is the recommended phase 2 dose. And they also looked at characterization of clinical efficacy, pharmacokinetics of Remidusib as part of the secondary objectives, including biomarkers, which looked at T cell expansion within the body. So what they actually did was they gave lymphodepleting chemotherapy, prepared the T cells, CAR T cells, and then these CAR T cells were given at the dose of 5 × 10⁶ cells per kg of the patient's body weight. In addition to these CAR T cells, 1 week after the CAR T cell infusion, Remidusib was given at the dose of 0.4 milligram per kg. Here there was a dose escalation. Patients either received single dose of Remidusib or they went on to receive up to 30 doses of Remidusib in the 3+3 study design. So 8 patients' data has been reported. These were patients that were recruited up to September 2022. All 8 received the CAR T cell dose. 3 patients received, in addition, a single dose of Remidusib, and the remaining 5 received multi-doses, which ranged from 1 to 30. So since this was a phase 1 study and looked at safety as the primary endpoint, these were the major toxicities that were noted. So the most common toxicities that are expected are myelosuppressive events because of the lymphodepletive therapy before you give the CAR T cells. But in addition, there are certain toxicities of CAR T cells that are of importance, like the cytokine release syndrome, which is a known toxicity of CAR T cells and was also seen in all patients here. Most of them were grade 1. Only 2 patients had grade 3. Immune-related neurotoxicity syndrome, also a known toxicity of CAR T cells. Only 2 patients had it, one grade 1, one grade 4, but both resolved. And there was one death because of neutropenic sepsis, which was considered a dose-limiting toxicity. So this was the secondary endpoints looking at the activity of the cells. So they had a PSA 50% response at day 28, that is a decline of PSA to 50% of the levels in 3 patients out of 7. And these are the data available till now on the RECIST responses of these patients. So 1 out of 8 had a partial response, 3 had stable disease, out of which 1 had a longer duration of stable disease for more than 9 months. 1 progressed, and 2 patients had not yet reached the imaging time point in the trial. And as they looked at biomarkers and the activity of the T cells within the patients and how they expanded, these were the T cell expansion that happened. So these were the copy numbers of the T cell in the first week. And when they administered multiple doses of remidoside, which stimulated the T cells further, these were the re-expansion copy numbers. So the authors concluded that this product, that is the T cell product that they have designed, targeting PSCA, had good evidence of biological activity and known toxicity like other T cells previously reported. Also, they said that remidoside, which they are administering as part of the trial, induced activation and proliferation, and they were planning to escalate further to increasing the dose of remidoside to more than 0.4 milligram per kg, and also escalating the CAR T doses. But there's an update. This study now is, although ongoing, has stopped recruiting patients further because of the dose-limiting toxicity. It is not very clear whether it is the same death due to FN that was the dose-limiting toxicity due to which the study has been, the recruitment has been stopped, but the final results are awaited, and this current data is available only in the poster form. So my conclusion would be the take-home message to learn is that definitely CAR T cell therapy has promise in prostate cancer. 3 major molecules that are being evaluated, including PSCA, PSMA, and EpCAM, are likely to go on to phase 2 and other phase studies in the future. Whether they're going to come up to routine clinical practice, right now it's too preliminary to say. There need to be more studies both to establish the dose and probably also safety issues that are associated with these CAR T cells. But having said that, this could be a potential new therapy for patients who have no other options in metastatic CRPC. So thank you, and I hand over back to the chairperson.